Okay, in this video tutorial we're going to look at a couple of different types of cell divisions called mitosis and meiosis, and you probably had a look at these as juniors. So starting with mitosis, mitosis is really a really key process that is necessary for growth and repair because it produces more identical cells. So for growth and repair, or regenerating, replacing damaged or dead tissue. Mitosis is also a mechanism for reproduction in some organisms. So those things that reproduce asexually, so simply like plants that grow by budding, or bacteria, things that grow by binary fission, essentially reproduce simply by a process of mitosis. Now, a key thing in this process is that the resulting daughter cells are identical, so the DNA must be the same. So a lot of care must be taken there. So these cell cycle, mitosis is only a small part of the cell cycle. So if we've got a growing population of cells, those cells are going essentially through this cycle here. And it's worth noting that even in a growing population of cells, most of the cells will be in interphase, the in-between phase. And interphase can be split into a G1, gap 1, S, and G2, gap 2 phase. And basically during the gap 1 phase, the cellular contents are growing. So the cell is making more proteins. Its organelles are either growing or increasing in number. So some organelles, like the endoplasmic reticulum and I think the Golgi apparatus, simply increase in size, whereas others may increase in number, like the mitochondria and the chloroplasts. So some of them during when the cell actually ultimately divides are literally divided, are split in two, and some, if there are multiple copies of it, you'll get some in one cell and some in the other. And it's worth noting, I think the reason for that is if you look at organelles where you get multiple of them, so things like the chloroplast and mitochondria, they actually have some of their own DNA, and so in that instance the chloroplast and mitochondria, we think they were actually at some point prokaryotes that were engulfed by a larger eukaryotic cell and have evolved a symbiotic relationship. So they're almost like a cell within a cell, really. But anyway, that's getting into a bit too much detail. So here the organelles are grow or are copied, are duplicated, and so that ensures that when the cell divides, we don't just get cells that are smaller and smaller and smaller. So this is kind of growth of the actual cell. S is for synthesis, so this is where the DNA is synthesized, or basically this is DNA replication. So we've looked at that too. This is where the DNA is actually copied. And in the gap 2, basically there are checks to make sure that the DNA was copied correctly, that we don't have any mutations. Usually some mistakes are made, and so at this stage they're corrected. And there's also a few other sort of cell signaling checks there to ensure, that the cell should go through with the division. Then we get into the mitotic phase, and there's two parts to that. Mitosis, which itself is really just the nuclear division. So mitosis is really talking about the nuclear division, and then cytokinesis, the cell division, and we'll look at those in a second. Now there's a little branch off to the side here, and that's because a cell can go into cell cycle arrest, so it stops growing essentially, so it leaves the cell cycle. So this is the cell cycle for a kind of a growing population of cells, when the cells are actually growing. So if we look at mitosis, you've probably covered mitosis as a junior, and so it's I'm just going to go over the basics quickly, and then we'll look at it in a little bit more detail. So mitosis, really the key thing is that what you need to realize is you start off here with the normal number of chromosomes, which is actually 2 of each. So we often say this is diploid, or 2n. And the first step in any kind of cell division is actually DNA replication. So that's nice and easy to remember. It's always DNA replication. Now you can see here in a human cell, we would actually have 46 chromosomes. We've only shown 2 because the diagram gets a bit complicated with 46. At this stage, you actually still have 46 chromosomes. It's just that each chromosome consists of 2 copies, or what we call chromatids. And this is usually when you can actually see them under the microscope. and that's when it's the best time to take a picture of them, or when you can actually see the chromosomes. And if you take a picture of the chromosomes under a microscope and arrange them according to size, we call that a karyotype. And so often that's done to look for defects. So in the old days, some poor sap had to literally cut out all the chromosomes, and the chromosomes could be stained, and they'd show sort of a banding pattern, and that would help scientists to kind of pair them up into their 23 pairs, and then they could look for if there were any defects with them. So the key thing to remember from mitosis that will help you remember the difference between mitosis and meiosis is that if you struggle to remember this kind of sequence, is this bit here. So DNA replication, the first bit's the same for any kind of cell division, but for mitosis, the chromosomes line up end to end. So they line up in a row like this, end to end. And then the chromatids are simply split. The chromosomes are split into their chromatids. The chromatids are pulled to each end of the cells by spindle fibers, and then the cell actually divides, and you're left with daughter cells down here. And so the daughter cells down here are identical in every way to the parent cell we started with. So the key points for mitosis, I've put those just down the bottom here, so we'll zoom in a little bit there. The purpose of mitosis is for growth and repair. It occurs in the somatic cells, so the skin cells, and it results in two daughter cells that are genetically identical. The daughter cells are diploid. They have the normal number of chromosomes, or two of each. So it's worth noting, so in a human cell, we have 46 chromosomes is the normal number, or those can be arranged into 23 pairs, and we'll talk about that later, and two of these are sex chromosomes. So 44, the rest of them are what we call autosomal chromosomes, which just means those that aren't involved in sexual determination, and then two of them are sex chromosomes, and those are the X and Y chromosomes. So those are the key points for mitosis, and we'll go into a little bit more detail now. So moving down. Okay, so here are the key phases of mitosis, and I'll just zoom in a little bit. Oh. There we go. So during the first phase is prophase. So interphase is that kind of in-between phase where the DNA is actually copied. And then we go on to prophase as the start of the actual nuclear division. So during prophase, the chromatin condenses. Chromatin is that material, the protein plus DNA, into chromosomes. So what's happening is the DNA is being tightly coiled around those proteins, those histones, and that's being coiled up tightly into those chromosome structures. So at this stage, the chromosomes actually become visible under the microscope. At this point, the centrioles also move to the opposite poles and start to form spindle fibers. It's also at this stage that essentially the nuclear envelope, so you can see a nucleus here, just move that over a little bit, you can see a nucleus here. The nucleus here around the chromosome starts to break down. So you start to see the nuclear envelope or membrane, and the nucleolus will have disappeared, and so you kind of don't have a nucleus anymore, a defined nucleus. The next phase is called metaphase, and during this phase, the chromosomes line up in their homologous pairs across what we call the equator of the cell. So if you think about the equator of the planet, it just means across kind of the middle of the cell. And so you often can't see them very clearly, they're sort of splodgy kind of, but they're all in the middle there. You can see they're all in the middle there. So they attach to the spindle fibers. So the centrioles have moved to the opposite poles during prophase, and in metaphase, these chromosomes are attached to the spindle fibers, and then we go on to anaphase. So during anaphase, the spindle fibers shorten, and the chromosomes split apart into chromatids, and the chromatids are pulled to each end of the cell. So those chromatids are pulled to each end of the cell. And this basically separates the chromosomes into their chromatids, and it means that a copy of each chromosome, those chromatids, will end up at each end of the cell. So we then move into telophase, where the chromosomes have reached the poles now, and basically those spindle fibers start to break down, and a nuclear membrane starts to form around the chromosomes, and a nucleolus, a dense patch within that, starts to become apparent as well. And so the reason you lose that chromosome structure is what's happening is basically it's reverting back from that chromosomal state back to that kind of nuclear state where you've got a nucleus with a nuclear membrane, and inside that the chromosomes have uncoiled or relaxed slightly. So the chromosomes uncoil, although you maintain sort of a more dense patch, or a dense patch forms called the nucleolus. And the last stage, cytokinesis, is often sort of kept separate from mitosis, and some textbooks include it as part of mitosis, is essentially the actual cell division. So this is when the cytoplasm and the cell contents are split into two. So the cell membrane actually pinches in, and the contents of the cell are split in two, and each of the resulting cells will have their own nucleus, and they'll grow in size to essentially then be identical to the cell we started with. So there we go. That's mitosis and the cell cycle. So it's not the only type of cell division. Mitosis, remember, occurs in any body cell and is really just about making identical or copies of the daughter cell. So one way some of my students have remembered that in the past, they'll say mitosis occurs in your toe. So if you cut your toe, or when you're growing, mitosis is occurring in your toe. So the other type of cell division is called meiosis. And so why do we actually need another type of cell division? Well, it's what contributes to sexual reproduction. So in animals that reproduce sexually, we have adults that both have 2n, a diploid number of chromosomes. That means they've got 2 of each chromosome. Now, if they produced sex cells, eggs and sperm, through mitosis, then the eggs and sperm would both have 2n, the full number of chromosomes. And when they join together, the zygote, or the offspring that results, would have double the number of chromosomes. And in fact, the number of chromosomes would double every generation. So meiosis is often called a reduction division, because it reduces the number of chromosomes from 2n, the diploid number, where there are 2 of each chromosome, to n, the haploid. So this is diploid, di for 2, so diploid, the double number, or the normal number, 2 of each chromosome. We've got 2 versions of each chromosome. And if you think about it, the 2 versions of those chromosomes, so if we put them in a karyotype, organize them according to size, we've got 22 pairs of chromosomes and 1 pair of sex chromosomes, so 22 autosomal pairs and 1 pair of sex chromosomes, and 1 member of each pair. So if you look at chromosome 1, we've got 2 versions of chromosome 1. We inherited 1 of each of those from our parents. So in the egg and the sperm cell, meiosis ensures that the egg and the sperm receive 1 member of each of those pairs. So it has an N, it has the haploid, or 1 of each chromosome, rather than 2 of each. So you have the haploid number of chromosomes. So those fuse together to form a zygote, which is at this stage a single cell, but it now has 2 of each chromosome again. 1 that it's got from the father, that's the mother, the egg, and one of each chromosome from the father through the sperm. So it now has 2 of each chromosome, 1 member of each pair has come from mum, and 1 member of each of those pairs of chromosomes has come from dad. And that will essentially grow into a juvenile and continue to grow, so those are mitotic divisions, or mitosis, into an adult. And then again, meiosis to produce the sex cells, those fuse, fertilization, you get a zygote. Mitosis is what causes the growth, and you go round through that cycle. So this is kind of the reproductive cycle for any organism that produces sexually. So why produce sexually? Well, sexually produces variation in the offspring. And there's several reasons for that, and I'm not going to go into all of them. But in part, if for no other reason, one of the key reasons you get variation is you've actually got a new combination of genes from both mum and dad, from two organisms. So the fact that the offspring is already has contributions from mum and dad mean that basically it's already going to be genetically different. And genetic difference can be an advantage to a population because it means that in the population we have greater genetic variation. And that plays a really important part in evolution, but also in the survival of a species. The greater the genetic diversity or variation in a population, the more likely it can cope with environmental change. Because if the environmental conditions change, there'll hopefully be some individuals in the population who are adapted to the new conditions. So let's have a look at meiosis. So meiosis, again, we start the same starting process. We start here with DNA replication. We copy the single sausage chromosomes into their double sausage versions. But the key difference here in meiosis is that the chromosomes pair up side by side, and we call those pairs homologous chromosomes. So in a human cell you would see 23 pairs of chromosomes. The largest pair we call chromosome 1, and we've got two chromosome 1s that pair up. The next largest pair is chromosome 2, so those are again we've got two copies of chromosome 2, and each of those pairs, so for instance here the red one, that red chromosome here may have originally from here come from our mother, and the blue chromosome may originally have come from our father. So all we've done is taken those chromosomes and turned them into the double sausage version. They've replicated so that they have a copy attached. So the thing that's really different in meiosis is that the chromosomes here pair up side by side. So instead of pairing up end to end, so it's at this point already here you can see that those chromosomes are now being separated. So normally in mitosis, because they line up end to end, each cell gets a copy from every chromosome. Whereas here, the two chromosomes here, if these are both chromosome 1, we're only going to get DNA from one of those in the daughter cells. They're already separating. And so this is really important because we have two versions of each chromosome, one that we've inherited from mum and dad, we often have two versions of each gene. So for instance, the genes for, say whether you can roll your tongue or not. So maybe from your father, on this chromosome here, maybe you inherited right at this point here, you inherited a gene that enables you to roll your tongue. And from your mother, maybe you inherited the non-tongue roller gene, and that will be found on the exact same point on that chromosome, because these chromosomes are almost identical. They're almost identical. They contain slightly different versions of the same gene. So these chromosomes contain what we call alleles. And you've probably covered those, or you should have covered those in previous years already. So alleles, the definition of an allele is simply an alternative version or form of a gene. Alright, so these chromosomes, the sequence is almost identical on these chromosomes, the one we got from mum and dad, but in some of the genes within those chromosomes, there might be just one or two bases that are different, could be very subtle differences, and that might make this gene, in this case maybe non-functional, so it doesn't help you roll your tongue, and in this case it helps you roll your tongue. So if you get two different genes from mum and dad, what happens? You can roll one half of your tongue? No. Alright, so this is the idea, is one allele, one version of the gene will be dominant. So in this case, if you get one copy of the gene that enables you to roll your tongue, then you will be able to roll your tongue. So here we've got a single gene, and we've got another version of that gene that could be the same or slightly different. And if they're different, one of them will be dominant, one of them will be recessive. But when we're looking at producing sex cells, you can see at this stage already we're going to separate those versions, those alleles, those versions of a gene, and it means that we're going to pass on to our offspring. Either we're going to pass on the version we got from dad, or the version we inherited from mum. And this is how you can pass on, if this is a recessive allele, one that was hidden, this is how you can pass on a hidden characteristic to your children, and why they may have characteristics completely different from yourself. So the key idea here in meiosis, the chromosomes line up in homologous pairs side by side. The pairs separate, and then there's a second division where each of those chromosomes is split into its chromatids, and we end up with 4 daughter cells. So the purpose of meiosis is to make gametes, or sex cells. So in humans, that's sperm and ovum. Where does meiosis occur? It occurs in the testes and ovaries. And it starts in specialized cells called germ cells. So that's often a trick I've seen examiners use. They might say, where does meiosis occur? And students will often say, in sex cells. Well, no, sex cells are the product. They're not where it starts. So just be aware of that point. So it produces 4 daughter cells, and they are genetically different because they will get, some of the cells will get the version of say chromosome 1 that we inherited from our mother, and some of them will get the version of chromosome 1 that we got from our father. So meiosis ensures that our cells have 2 of each chromosome, and our meiosis ensures that our sex cells will get 1 member from each of those pairs. So a sex cell, a sperm and an egg cell, always has a chromosome 1 and a chromosome 2 and 3 and 4, and those always have certain genes on them. And that ensures that when it fuses with the complementary type of cell, whether it's a sperm or an egg, that the offspring will also then have 2 of each chromosome, rather than having 4 of one and none of the other. So we have 2 of each chromosome in our body cells, and meiosis ensures that our sex cells will contain 1 member of each of those pairs, 1 of each chromosome, so half the number. So if we look at meiosis in a little bit more detail, and you don't actually really need to know any more detail for the Cambridge exams, meiosis is just be aware, like as I mentioned earlier, it can be called a reduction division because it reduces the number of chromosomes. But it's worth just mentioning that essentially the phases are the same as they were in mitosis. So we start off with interphase, that in-between phase where the DNA is copied, prophase where the chromosomes condense, metaphase where they line up, except this time in pairs, in those homologous pairs, anaphase where the chromosomes are pulled apart, telophase where the nuclei start to reform, and then you get a cytokinesis where the cells actually separate, and then the whole thing starts over again. So you get a prophase 2, metaphase 2, anaphase 2. The only one that's left out is the interphase, the in-between, and that's because the DNA doesn't have to be recopied. You don't have that big gap, that big waiting in between. It goes straight into prophase again. So it's kind of just a double division. It occurs twice. So you don't really need to know the phases for meiosis, but I figure, they're basically the same. It's worth mentioning at this stage. So, just to recap, mitosis, is a division for growth and repair, produces genetically identical diploid daughter cells. Meiosis produces daughter cells with half the number of chromosomes. So you can see that here. Not so clearly in this one actually, but this original cell had 4 chromosomes in it. You can see 2 red ones, 2 blue ones. And down in the daughter cells at the very end, they'll each have the 4 daughter cells. They haven't quite split here. Will each have 2 chromosomes in them. And so, as I said, meiosis ensures that. So normally our body cells have 2 of each chromosome, Mitosis ensures that they have an identical, an exact copy of that. Meiosis ensures that we produce sex cells, sperm or egg, that only have one of each chromosome in them, so that when it fuses with the other one, it will have a full set. It again will have two of them. And the reason why, if you have children, that even if you have children with the same partner, the reason they look different is every time you put together an egg or a sperm cell, it's a little bit of a lottery essentially for which member of chromosome 1 will end up in the sperm cell, which version of chromosome 2 will end up in the sperm cell, which version of chromosome 3. Every time it's a bit of a lottery with that. So are you passing on the version of chromosome 1 that you got from your mum or dad? Are you passing on the version of chromosome 2 that you got from your mum or your dad? Every time it's a lottery, and so you get a different kind of combination being passed on each time. So I hope you found that helpful. That's basically an introduction to mitosis and meiosis and the cell cycle. And in the next video, we'll have a little bit of a look at what happens when that goes wrong.